talk is by Dr. Eric Tkaczyk. And Dr. Tkaczyk is a physician scientist with research interest in biophotonics for diagnosis and treatment of skin disease. He's a graduate of the MD PhD at University of Michigan, with PhD in electrical engineering. So very unique person that's really interdisciplinary, able to merge together the medicine, the engineering, and dermatology, as a matter of fact. So Dr. Tkaczyk is active in the leadership of a number of international groups and conferences related to artificial intelligence and dermatologic imaging technologies, especially applied in the post-transplant setting. So thank you, Eric. Yeah.
Well, thank you so much, Steve, for the kind introduction. It's such an honor to be at this symposium and to have a role in this just wonderful collaborative community around GVHD. And it's especially been very motivating for me personally to meet Jerry and Meredith and patients and their families, because it keeps it in perspective. It's very humbling. And I also want to emphasize that my collaborators, I've put some photos and names of some clinicians, wonderful clinicians who have taught me so much, as have patients and their caregivers, and motivate us to try to make a difference. And I'm just learning the slide. Oh, here we go. Okay, so as a dermatologist, it struck me how important the skin is in chronic GVHD, and is what attracted me to really apply my efforts to this specialty. Because as you know, it's affected most of the time. More than 75% of patients with chronic graft-versus-host disease will have skin involvement. And it's very striking how much morbidity it can cause with the altered appearance of the skin, the itching, the pain, the wounds, the restriction even of breathing. So it can be quite dramatic, or it can be less dramatic and subtle and maybe not be affecting so much quality of life, but it's still very important as an endpoint and as a diagnostic window into the patient's overall GVHD, because it's so visible. The involvement of the skin is associated with mortality and other very important outcomes. And so it's prominent in the staging and response, and importantly, the assessment of new therapies. So the goal of that I have with my collaborators is to develop and implement technologies that do very rigorously, in a measurable way, quantify what we try to do in clinic when you send a patient to a GVHD dermatologist, which is we look at the skin, we touch it, and sometimes we biopsy it. We try to do this with technology. Today, the talk will focus on the analysis of what we can see with the artificial intelligence analysis of photos, but we also have very active lines of research using measurements of sclerosis, which some of you have seen. I've been carrying around this little Myoton device that we use to track sclerosis. And then I've also told you about several of the attendees about this microscope we have, where we get this non-invasive biopsy, hopefully as a source of biomarkers, but focusing on what we can see. So here's the first photo. It's not the first photo, but it's the first patient who ever consented for one of our studies and took a photo of. And just like me, this is Mr. T. I used to be Mr. T, now I'm Dr. T. But here's a photo of Mr. T's back. And I remember very vividly this patient encounter where I had just started as a young faculty, and I was there with my mentor, Madan Jagasia, in this visit. And the patient wasn't doing so well. And so Madan offered him several trial options. And it was very surprising to me then, and equally surprising to me now, that the standard of care for patients with very severe chronic GVHD is enrollment on a clinical trial. And so at the time, we were enrolling for the pivotal ibrutinib trial, which around the time Ed Cowan visited us to train us in skin scoring, got, thankfully, FDA approval. And one of the enrollment criteria for that pivotal trial in ibrutinib was having more than 25% body surface area of erythema. And so here's the photo of this patient. So I'm going to do an audience participation here without technology, just show of hands. Those of you at home, I can't tell if you're raising your hands, but please do anyway. It's good for your blood flow. So this patient, are they eligible for the trial? So I'm going to ask you, if you assume that the rest of the body is equally affected as this in this photo, would this patient have under 25% body surface area, in which case they would not be eligible? And that's question A. And I'm asking if anyone could raise their hand is where you've been. Okay, we have 1, 2, yes, 3, 4, 5. Okay, so several experienced transplanters looking at this photo would judge this as under 25% body surface area. What about over 25%? B, and getting a few more hands, and over 50%. Any takers? And is anyone unsure? A few are unsure. Many are unsure. Okay, I was also quite unsure at this time. And so that's actually why we flew Ed Cowan out to train us in how to estimate the body surface area. And it turns out that even if you're a dermatologist who's very engineer-like, like me, focused on details, you need a lot of training to do this well. So I think one of the key messages of my talk here is, can we stop asking our doctors to do computing and have our computers do the computing for you? And so I am going to get to the answer of this for that question in my talk. So the outline of my talk is, first, I'm going to talk about the importance of accurate assessment of chronic GVHD and the challenge of estimating BSA, the possibilities with AI and other technologies, and especially this challenge of expert variation, which is under-celebrated but very important. And then I will close out by giving you some suggestions just from our trial experience on how you can get some great skin data in GVHD trials that you're planning now while you're waiting around for the engineers to get things going. So the current standard for evaluating GVHD is the NIH skin score, and it really has been a landmark for the field. The NIH Consensus Development Project, which many of you, notably Dr. Pavletic here in the audience, have been very pivotal in, because part of the attraction for a dermatologist to graft-versus-host disease is because it really replicates an entire textbook of dermatology. It's almost every condition of dermatology. GVHD can look like almost any condition in dermatology. And so that makes it very hard to evaluate, because it's not like psoriasis where you can recognize it from the doorway and mark it out sharply. And so the NIH Consensus Development Project really did a lot for the field by dichotomizing the findings and developing a way to consistently stage them. And so the NIH skin score divides the findings of cutaneous GVHD into erythema, which is the visible epidermal change, and sclerosis, which is the fibrosis that can be in the dermis or deeper. And so about 80% of patients with skin GVHD have epidermal changes that are visible as erythema. About 40% have these sclerotic findings by our analysis of the consortium. And so it's more than 100% because there are many patients who have both findings. And so here is the NIH scoring system. In 2005, in the original scoring system, Ed Cowan had developed these scoring sheets, which he taught us to do as well for very rigorous scoring. But it turns out it's very hard to implement this rigorous scoring where you go through each body region and you estimate the surface area. So in 2014, it was revised to simply bin the surface area of erythema into 3 bins of involvement or no involvement. And sclerosis, you just check off, is there deep sclerosis? Is there superficial sclerosis? And that's how the NIH skin scoring is done. And one of the great data sets we have back when people were putting a huge amount of resource and effort into doing that body surface area estimation is the Chronic Graft-versus-Host Disease Consortium, which many of you in the audience have been highly engaged with. Thank you for all your efforts on scoring the skin on these patients. So we have body surface areas and the 2014 NIH score on several hundred patients from this 11-center consortium. And my collaborator, Emily Baumrin, whose photo you have there, together with a medical student, Laura Baker, now dermatology resident, and Stephanie Lee, we looked at some of this data because we tried to see how much prognostic value does the skin have. And this had been done before. It's been known for more than probably 10 years before I even started studying GVHD that the reversal of erythema is associated with survival and other important outcomes. But our interest was to quantify that association and to actually put some numbers behind it in a rigorous way. So we looked at the 206 patients in the consortium who had erythema at their first visit and had a follow-up, and we measured the prognostic after many statistical consultations with several center statisticians. We measured the prognostic value of the skin involvement surface area by looking at the likelihood ratio test statistic. It's a mouthful, the likelihood ratio at the first follow-up. And we looked at the association with several outcomes, most notably non-relapse mortality, which, as you probably know, is a surrogate for death due to graft versus host disease. And so the likelihood ratio for this body surface area of erythema was about 18 in this cohort, but for the NIH skin score, it was only 9. And for other organs, it was under 8. And so the higher the likelihood ratio, the more prognostic value for predicting the mortality, the non-relapse mortality. And so interpreting this data, it means that it's actually a linear interpretation. You have double the prognostic value of BSA that you have with the NIH skin score. So when you do an NIH skin score and you categorize the amount of surface area, you lose a lot of information. And so your prognostic value is cut and down by a factor of 2. And this can be illustrated if you look at some traditional Kaplan-Meier curves or at cumulative incidence curves, which I have a couple illustrated here. So on the right, you can see the cumulative incidence of non-relapse mortality for patients who had more than 10% surface area involved versus under 10. You can see a wide separation, but if you look at NIH skin score, greater than 1 or just 1, then you can see that there's less discrimination because you're sort of losing information. But I don't advocate thresholds. What I advocate for is, can we find a way to recover that informative body surface area in a practical way? Because it's not practical to have your clinicians computing. But the point of this slide is that there is a lot of value if we could measure the surface area well. And part of the impracticality of measuring surface area and why justifiably the 2014 consensus stopped doing it is because there's a huge error in measuring surface area. And so here's a study where Dr. Cowan had flown to many centers and trained people to assess the surface area. And here are the errors that you see. So on this plot, on the x-axis, you can see the surface area that Dr. Cowan assessed as active with erythema. And on the vertical axis, you can see the error, not the surface area, but the actual error in measuring surface area of the trained skin scores. And you can see that in cases, the median error was around 20%, but then you can see in some cases, the error was as high as 80% of the total body surface area that the scorers who had been trained were off by. And so even though measuring the extent of skin involvement is very important, for example, in the ibrutinib trial entry criterion, it's very challenging in practice because the error is often more than that 25% entry criterion. And also in some studies, it's been reporting that scoring time is reported by providers to be a barrier to many of them for even doing the 2014 NIH skin scoring. I want to make you all feel a little bit better about yourselves because I don't want to make anyone feel like transplant providers or skin scorers aren't trained well. I think you're trained very well. The problem is actually much beyond transplant. This is a general problem in the skin. It's a problem of the skin. It's a problem in dermatology. If you look at psoriasis, there's a several-fold variation in the surface area, which is a very much easier disease to score. Several-fold variation in surface area that dermatologists are estimating. So here's a study where this boy was examined by many dermatologists, and they were asked to report how much surface area is affected with psoriasis. Very well defined, very obvious. Even an elementary school student could tell you where the psoriasis is and where the skin doesn't have it. And yet the dermatologists had a range of several-fold. And their median surface area, they reported it was 27%, which I think is our magic number today. But at the same time, when they traced out in cellophane, they actually measured the exact centimeters squared affected. It was only 5% for this boy. So dermatologists, experts at examining the skin, asked to look at only one disease, only one organ, can't do it very well. And then in the meta-analysis from the burns literature, this was consistent as well. Burn surgeons, when they looked at referrals, asking to, very important in burn, the fluids are given based on the surface area of the burn, and the referring centers to top tier burn centers were almost always overestimating, and it would have resulted in inappropriate fluids, inappropriate management of burn patients more than 75% of the time. So we're in good company with our problem. We have a very important measure that we need to measure, which is the surface area of affected skin in cutaneous graft versus host disease. But we know that it's a lot to ask a human being to do it, and human beings can't really do it all that well. But because we're in good company, a lot of other people have been working on this problem. And so I think the answer often lies in just looking to what other people have done to solve their similar problems. So in summary, this is the first part of my talk. In summary, measuring erythema and the surface area of it is very important, but it's difficult. And if we could get the surface area accurately, we could probably double our prognostic value compared to our NIH skin scoring. But humans are not good computers of BSA, but maybe computers are good computers of BSA. So that's the second part of my talk. And here I'm going to talk about our efforts to measure erythema from photos. And I'll talk about our 3D photography efforts, our deep learning, artificial intelligence, the problem of expert variation, and then some new imaging technologies, which hopefully will help us fight this. So here's a commercial 3D camera, the Vectra camera from Canfield, which I bought in 2017 to start our GVHD program. And it's actually the camera I used to take the first photo I showed you. And here you can see the raw image, but it's actually, the camera has two eyes. So it can see stereoscopically like a human. It's all calibrated, the color, the flash, everything's very standardized with this camera, commercially available. Anyone can buy it. I've bought several like this. And so on this photo, I went into the 3D software and I just marked out the area that I thought was affected. And I got the answer was 27% in this particular case. So for those of you who answered A or B when we did the audience poll, you were pretty close to what I said because it would have been a pretty borderline call. Of course, the question is who's going to do all these markings? That particular photo took me a couple minutes to mark up, and that's only one portion of the body, so not practical to have your clinicians using that software probably themselves. But fortunately, again, looking to problems that others have solved, with the explosion that you've probably heard about of deep learning and image analysis, for example, in self-driving cars, there's a lot of technology that's out there at the level where even some high school students know how to implement it, to have automated AI-powered image analysis. And so the way it works is you basically take an off-the-shelf neural network, which is designed to sort of simulate a brain's visual cortex. There's different layers of neurons. These are digital neurons, and they have connections, and the strength of the connections is modulated after it sees a number of examples. And so you can get a neural network off the shelf. You can download it from repositories, and it's seen 2 million images of flowers and cars, and it knows how to recognize a lot of stuff in photos. But it hasn't seen that many GVHD images. So our task has simply been to take a brain, a neural fake brain, artificial intelligence brain, and just train it to recognize GVHD, much like we train our skin scores. And so with our 3D photography, with a small database, we're able to do some technical tricks like data augmentation and rotations, and we can show repeatedly this algorithm, we can show it these images, and then it assigns a probability of GVHD to each pixel, and we can get this, what's called a segmentation. It can mark out the areas. And so in this paper that we got out last year, here's an example on that same photo where our algorithm, which had never seen this patient, we'd held this patient out from training, where in blue, our algorithm's marking out where it thinks the borders are, and in green are the borders marked out with the opinion of our Vanderbilt sort of training school. And if we look at 300 images here and this box plot on the right, you can see that the median error when we measure the number of the fraction of the skin that it was off was about 9% on these images, which is more than twice as low in errors as the skin scorers who are trained to do GVHD scoring. Unfortunately, we trained on 36 patients' images. We had about 700 images for training. And then we look at the performance on held out patients. In the light-skinned patients, the performance was actually really, really good. But when we look at the dark-skinned patients, of which we only had 3 in this population, in this cohort, then the error was significantly higher, 24% error, which is about the same as you would get with a clinical exam. So we need, in order for this technology to be equitably implemented and robust, we need a lot more photos. So if anyone has any photos they'd like to set up a transfer agreement on or help us take, it'd be great for you to email us. And also, we trained with these 3D photos, but in the real world, you're much more likely for your patient who you're measuring to be taking photos with a smartphone. So we're also looking at what's called this domain shift problem in artificial intelligence, where you're trained on one specific technology, and now we're trying to work in another one. And this is common, for example, when CT scanners are switched in a hospital, AI stops working. And so, but it seems promising. Here's a photo that Mary Flowers and Mishi Shinohara had kindly sent from Fred Hutch, and this is our algorithm. This is the first 2D photo the algorithm has ever seen, and it seems in blue, it seems to be marking out pretty well compared to what a dermatologist marked out. And so here, I'd like to take a little brief interlude to talk about a great challenge here, which is we've trained this algorithm to mark the way I feel like my team should score GVHD after Dr. Cowan had visited us and trained us to do it. But actually, there's several GVHD dermatologists out there, and there's a lot of transplant physicians who might score differently. And this really came to a crux when we assembled several of our colleagues, mostly dermatologists like Julia Lehman from Mayo Clinic, Dr. Cowan from NIH, Mishi Shinohara, and Mary Flowers from University of Washington. And we were marking out these photos in our calls. And I said, okay, let's see how we do it. Let's just all mark the photo the same way we're asking the AI. And here are the results. So on this image, this is a Mayo Clinic patient from Dr. Lehman. And on the right, you can see us marking out individual pixels that someone identified. So if the pixel's red, it means that actually Mary is a transplanter, as you know, but the rest are dermatologists. But red means that 4 out of 4, all the experts marked it. Blue means that only 1 of the 4 marked it. And you can see this huge variation. So this was obviously not good to train the algorithm on. So we spent more than a year on these weekly, not weekly, but periodic consensus calls. And we'd all do homework. We'd share each other's photos. And after about a year, we finally got to some consensus guideline on how we should be marking. But without photos, you don't know this. When you're looking at your skin trial and you're looking at an outcome, if you don't have a photo of what happened to the patient, you actually don't know what people were thinking when they assessed this. So a key take-home message, I think, from my talk is take photos, lots of photos. And here's with our bioinformatics collaborators at Vanderbilt, we actually have built a web-based interface where you can go in. This is a published textbook photo from NIH, just for any of you concerned about PHI. But you can go in this web-based interface and you can mark out the areas that you think are affected. So I don't know when we'll be launching this study, but we're going to test these guidelines amongst experts, amongst patients, amongst lay people, hopefully. So if anyone wants to engage in that, we'll have a remote opportunity to help test guidelines for where GVHD actually is active. And a wonderful possibility that comes into play there is that now when you're taking photos in trials, if we have you can get multiple expert opinions on the patient who might not even be anywhere close to an expert, which is very important. And so now we're spending more time testing our algorithms against multiple expert opinions. So we're really penalizing the algorithm. It misses something that all experts would pick up, but we're not penalizing so much if it picks up something that most experts would ignore. So you can see our algorithm output compared to a 5 expert opinion here. Another thing that another reason I'm telling you to take photos is that I've really focused our time, first half of our time together, on the body surface area question. But we think that there will be new biomarkers from imaging that are very important. And so here I want to highlight redness. And with these photos, we were tracking this first patient's surface area. Here you can see on the top plot over time. So this is 9 months of follow-up, and you can see the surface area marked by different raters. And in that green curve, you can see that the patient's going from methotrexate to pentostatin. This patient eventually passed away from skin GVHD primarily. I mean, the skin GVHD didn't kill him, but he passed away from GVHD. And you can see that it's a flat line. The surface area that is marked here on these images, you can see from month 3 to month 6 isn't changing. But when we do a color analysis and we map out the redness coordinate, then we found that if you follow the redness of the patient, it's very much correlating with the clinical course. So that's just one example of an imaging biomarker that if we collect photos rigorously in a standardized way, we could go back and maybe even reanalyze data sets that you're collecting in trials. And to the problem of expert variation, new technologies could help that. Color could help with it. Really cool way to do color is to do the satellite sort of technology. If you have seen satellite maps where they identify vegetation or military compounds, they're using this hyperspectral technology where rather than doing like 3 colors like your monitor would have, they have 100 colors or 500 colors. And so we've done a bit of this with our GVHD patients, and then you can actually take these spectra. You can identify these unique spectral fingerprints, just like a chemist would do in their lab for different compounds. So here's an image, a close-up of a GVHD, of a patient with GVHD of their skin, and we've decomposed it where we've overlaid in red the hemoglobin distribution, and in black, we've overlaid the melanin distribution. And so here you see that we actually can get sort of an objective measure of the different components that are actually present in GVHD. So that's something we're pursuing to overcome expert variation. So in summary of our photo-based measurements, we can automate this process with artificial intelligence, deep learning algorithms, but we do need to test expert guidelines to implement it now. But in the future, color analysis, spectral imaging techniques may overcome this problem of expert variation. So our next steps are we need to assemble databases and we need to assess and address the performance in underrepresented skin types and prospectively validate these algorithms before we start using them. And finally, if you'll, I think we're a little bit over time, but if you'll humor me, I'll spend a few minutes giving some practical recommendations while we're waiting for these technologies to be validated enough to use them. Practical recommendations that I think you could implement in your next GVHD trials, which is the standard of care for your patients and for the patients in the audience. So first recommendation is to avoid mental math, have the computers do the computing. And one good way to do that is to use granular scoring sheets. Another recommendation is to use smartphone apps for taking your photos. Start with your baseline, take longitudinal photos. And then finally, for de-identification, consider standardized avatars. And I'll say a few details. So one of the studies that I've had a privilege to participate in a major role is the NATIONS trial. So this is a 20-some center study of drug rashes that put patients in the burn unit, Stevens-Johnson syndrome, for those of you who've heard of SJS. But in this drug rash trial, one of the lessons we learned is that, just like I mentioned with GVHD, just because you're a dermatologist doesn't mean you know how to score skin reliably within the trial confines. So training is absolutely required for all scorers, whether they're an expert or not. But the medical background is absolutely not required. So here's Dr. Phillips, the PI on the trial, and Madeline Marks, our study coordinator, who came from undergraduate with no medical background and scores basically on par with me after a little bit of training, is now training our other coordinators at 20 different sites to score this surface area of this drug rash. And so part of the way we train them is we have standardized kits we ship out with lesions that they tape on themselves, and then we know the exact surface area and we test them. And so here's a score sheet. And we test how they're doing, and we know if someone's off or needs more training. And then the score sheet is designed to avoid mental math. So we don't ask anyone to do any calculations. Those are all done automated in REDCap. What we ask the person to do is we ask them to look methodically at each region of the body and then tell us what fraction of it is affected. And so one thing that's very important and has really I've struggled with in the past is when I'm asked to estimate the surface area of the leg, because the leg is big and I have to look at the back of the leg, I have to look at the front of the leg. It takes me a lot of time and a lot of visual memory and mental math to estimate the fraction of the leg that's involved. So what we're doing for this trial, and we found has improved our reliability, is we just say, look at the anterior thigh. How much is involved? Half of it. Okay. Look at the back of the thigh. How much is involved? A quarter. You're done, and you move on. So that's actually improved our speed and accuracy. So the first recommendation was avoid mental math, use granular scoring, do your math in the computer. The second recommendation is to do standardized photo documentation with a smartphone app. Because in the melanoma community, it's so important to track individual lesions on a patient to diagnose the early melanoma and save lives. There's a lot of commercial vendors out there. We've collaborated with several of them on different trials. And so this particular one I'm highlighting that we're doing again on this drug rash study is Skin.io, but there's other options. But these apps on an iPad or an iPhone, they control the tilt, they show you contours, so that every time you're assessing a patient, a new one or a longitudinal assessment, you get the exact same body region. So your photographs are very standardized. And this raises a lot of possibilities to have a data bank of photos when you're doing a trial, because you can remove the space constraints and the time constraints of assessing patients. For example, in the burn unit for this trial, very, very rushed environment, because you can do it offline with the photos. It enables quality assurance, adjudication of cases, and testing new biomarkers like those color biomarkers in the future after your trial is closed, potentially exploratory analyses. And then, so what we've done with this trial is we've integrated that app that I showed you before, where we can mark out the areas that are affected, and the app that acquires the photos. So our REDCap actually shows the photo, it shows the score, it shows the marking that the study coordinator did on site. So this is an up and running trial now already. We've randomized patients already in open sites, and we're able to do this. So you can do this for GVHD. Transplanters are you have great systems for trials. I think there's no question this is achievable in the transplant world. And then finally, to the question of sharing data, because it's very important for progress to share your data, but sometimes with photos, it's hard to share it, even if it's not identifiable. So I'd recommend using avatars. So here's the Anatomy Mapper webpage. The CEO, Matt Melenda, gave me permission to share this with you, but this is something we've been doing for our GVHD patients. So you can see here, Anatomy Mapper has developed this custom interface that's specific for GVHD. you select the body region here, and you can map at any level of that region what you saw. If you see erythema, if you see sclerosis, if you see both. And then as a result, and I want to highlight Feng Yixi, who's a dermatology resident in the UK, who spent a year at Mayo Clinic analyzing a lot of photos, and you can see a patient with sclerosis and pigmentary changes and also active erythema here, who is being followed with this avatar. And this was actually presented yesterday in Berlin at the EADV, European Academy of Dermatology Venereology Congress, where you can have this standardized way of tracking your GVHD and potentially estimating in a more reliable way the extent of involvement. And this can be done anywhere. I want to emphasize this is very achievable now. We're involved with a monkeypox, Mpox now, randomized controlled trial called PALM-007 in the remote areas of the Democratic Republic of the Congo. Here's Dr. Inga Saknita from my team, who's here in the capital in Kinshasa, training the Congolese physicians to use the app and the photography systems. There she is on the embassy plane flying to the remote jungle area in Kola and the hot zone where it's super hot. And they're taking photos of these patients, very reliable photos. We've randomized more than 250 patients already on this trial and taken photos every day almost. And so in summary, to advance your GVHD trial skin assessment, I would recommend avoid mental math, test your skin assessors and train them, regardless of who they are, use photos, take photos, especially baseline photos, and advance the science by sharing your images or your time if you want to participate in some of our remote studies. So in conclusion, I hope I've convinced you that accurate skin assessment is critical in evaluating graft versus host disease and therapies, and that AI could help us by automating these measures, which humans can't really reliably do. And that smartphone photos and standardized scoring and apps can give you great skin data anywhere today and enable better technologies for your patients tomorrow. So I'd like to thank you. And if there's any questions, please, or anyone's interested in joining our team that you know, please feel free to follow up with my personal email. Thank you.